All right. Thank you, Levine. All right, let's try this again. So I need to share. I need to go to my window. All right. Okay, so we're back here of cytoplasmic extensions, the receptive surface. So what is the part receiving information? Some type of dendritic region. Okay, and for our multipotent neurons, or multipolar neurons, remember, I always think of it as like the head with the one eye, and that's the dendritic area. Now, you know with some of your bipolar and your unipolar that the dendritic surface might be free nerve endings, it might be some receptor, it might be something, and then there's kind of a long cytoplasmic extension towards the membrane. So not every neuron looks like the one-eyed monster, but in general, every neuron typically has many receptive capabilities. It can be talked to by multiple neurons, multiple signaling potentially, but it has one output. Okay, so in general, many things come to the neuron and influence it, give it graded potentials, and that neuron has one axon to make an output. And you turn it on by getting the action potential to go, or you keep it off if the action potential doesn't go. Okay? All right. I warned you that you would have to know your neuroglial cells, each cell of the central nervous system and the periphery, and then within, where we find each cell, what they do. So this question is asking you, which of the following neuroglial cells is within the central nervous system and produces myelin? In the CNS, it cannot then be a Schwann cell, right? Yeah. And it can't be a satellite cell. So Schwann cells and satellite cells are out because that is not in the central nervous system. What in the central nervous system is our myelinating cell? It's the oligodendrocyte. Okay? All right. Which of the following neuroglial cells is in the PNS and usually located in the ganglia region? So your dorsal root ganglia, your sympathetic chain ganglia, your parasympathetic ganglia. All right, that's not the Schwann cell, right? Because the Schwann cell needs to be located on the axon, and the axon is ending at the ganglia, and that's where the Schwann cell ends. You know, you don't usually see them there. So what are going to help protect the synaptic pieces and parts and any cell bodies in a ganglia? Satellite cells. Okay? All right. A neuron that conducts action potentials down its axon with the ultimate intent to trigger a skeletal muscle fiber. So is that a sympathetic neuron from the sympathetic autonomic system? No. Is that something from the parasympathetic system? No. All right. Is that going to be something related to sensory afferent systems? No, because they bring information too. So you're looking for something that says efferent, and you're something that probably says motor or somatic motor. All right, so those are like the buzzwords: efferent, somatic, motor. And what would be our neurotransmitter released onto a skeletal muscle? Acetylcholine. Okay, so that's why we're looking for motor or efferent neurons. Okay. In a neuron cell, the difference in electrical charges between the inside and the outside of a polarized neuronal cell membrane is usually negative 70 millivolts. So I'm giving you this number, and normally when we think negative 70 millivolts, is anything happening that we consider depolarization or hyperpolarization? No, that's its stable, steady state, kind of like something leaks in, something leaks out, and pump. Okay, so that negative 70, this is usually fairly stable, and the charge across the membrane is usually maintained at this value. Therefore, I am going to call this point in time, this value, if the neuron is sitting at that, I would say the neuron is showing its resting membrane potential. If the potential goes a little up, it would depolarize it. If it goes a little down, it hyperpolarizes it. What kind of potentials go a little up or a little down? Usually because ligand-gated channels open. Those are graded potentials. if an event happens where there's a rapid depolarization followed by repolarization, and everything is usually voltage-gated channels, and it's a repeatable, very much, it looks exactly the same over and over and over again event, what would we call that? An action potential. Okay? So that's kind of how you need to wrap your head around them. All right? Next question. We just kind of went through it. A shift in membrane potential to a less negative value from steady state. It's usually achieved in neurons by an influx of sodium. So if a positive ion is coming in, the inside of the cell is becoming less negative. So negative 70 would become negative 65, negative 60, negative 50, 55, you know, those things. That change from resting to a less negative value, we call that a depolarization. Anytime you have a depolarization and you come back to resting, that is repolarization. Anything from resting to a more negative value, hyperpolarization. And we went through, what are things that typically cause depolarization? Sodium comes in, calcium comes in, or chloride. Chloride could leak out, or phosphate, or something negative leaks out. Okay? How do I make a hyperpolarization? Potassium leaves, or chloride comes in. A negative comes in, or a positive leaves.
Isn't it usually like
usually
potassium going out, right?
Yeah, it depends. It depends. Okay. Next question. All right. If you put them in the wrong order, I had to manually correct this. So if you put potassium and sodium. Yeah. So what are the two major ions? The transmembrane potential, resting, graded, action potentials of a neuron are dependent upon movement of two major ions into and out of the cell. Name them. Sodium, and sodium is going to tend to be high outside, and it wants to come inside. Potassium is going to be the ion majorly inside the cell that wants to leave. Okay? Again, double check me when you check your quiz. If you put potassium first, I had to manually, because the auto grader didn't like that. Okay? All right. More key terminology related to these electrical things, right? Blank is a key term for neurons and electrical changes across the membrane. For an action potential, it is defined as the critical voltage. So this term can be applied a few different times and places. For an action potential, it's the critical voltage on the membrane that will trigger the start of ion influx by the voltage-gated ion channels at the axon-hillock segment. It is a critical voltage that will lead to propagation down the axon to the that should be synaptic terminals where the axon will release neurotransmitter. What is that critical thing? That is threshold. Okay? So that's that critical voltage where we start to see crap happen. If we don't hit that critical voltage, things can just kind of happen, but it doesn't necessarily mean the neuron's going to release neurotransmitter. Okay? The message may not be propagated. Repolarization of the neuron. So during an action potential, I have had rapid influx of sodium and I have rapidly depolarized. All of a sudden, I have a bunch of crap inside the cell that's positive, and I don't like that. So I want to repolarize. Repolarization phase of a neuron that has undergone an electrical event is usually the result of what ion moving out? Potassium. Now, does the cell really want to lose potassium? No. So towards the end of the repolarization, what is the cell going to start to really try to do? Use pumps to get rid of all the sodium and pull back in the potassium. But for a quick, within a few milliseconds, get a bunch of positive out, potassium is let go because potassium will flow. but then I need to pump things back because I don't want to keep sodium, because if I keep sodium, what am I going to do? I'm going to get big, because what's going to follow sodium? Water. And I don't want any exploding neurons, right? Because will they fix themselves? Not necessarily. Okay, the innermost layer of protection around the spinal cord and brain is the blank. The pia, right?
Yeah.
Yeah, I didn't hear anything. Okay, the pia. So what's the outer one? What's the middle? Subarachnoid. And then where do we expect to find fluid from the CSF? The subarachnoid space. Okay? All right, cranial nerves. Your friend's having their eyes tested. And one eye does not respond to dilation or constriction. So the iris muscle is not dilating or constricting, and the pupil's not getting bigger or smaller. Is that something related to the optic nerve?
Not the optic.
Not the optic. Is that something related to the olfactory? So it's not cranial 1 and it's not cranial 2. Okay? So we know that we have the oculomotor, abducens, and trochlear. But trochlear and abducens, do they play a role in dilation, constriction, movement in the eyeball? No, they just play one role with the muscle. So what is then the best answer? Oculomotor. Cranial nerve number 3, which is I. Okay? All right. Which of the following would not be found in your cerebral spinal fluid? Do you expect to have some glucose in there? You expect to have water? Do you expect to have some sodium, some potassium, some chloride? Yes, because we went through the neuron. Extracellular fluid has to be, high in sodium. So, what should we not expect? Should there be any red blood cells? No, and there should not be any white blood cells either, because the white blood cells could then start attacking my neurons. That's right, because they can go rogue and get crazy. All right, typical reflex. So you have a receptor. A receptor is going to be triggered. That receptor then has to get to the spinal cord. So what kind of neuron brings information to the spinal cord? An afferent sensory neuron. Then typically you have it synapsing at the posterior horn and an interneuron. One interneuron is going to the brain, but this other interneuron is going to be triggered because the threshold on it is hit. Because the stimulus is so powerful that enough neurotransmitter is released not only for the interneuron to go to the brain, but the interneuron to trigger this reflex. And it's going to need to move your hand away, move your foot away. So it has to be tied to the anterior horn where there sits your somatic motor neurons, your effectors. And it will trigger them, and then they will then release on the muscle or the gland, and you have that effect of pulling that limb away. There could be another interneuron, so if it's my foot, and if I have to pick up this foot, what does this foot have to be told to do? Take the body weight. So potentially, I could be talking to 3 or 4 interneurons, right? Who can be prepping the balance and the cross extensor, so I don't collapse down into the fire or whatever I'm pulling away from. Okay? So you got to have that pathway down. All right. Just like in our muscle organs, if we look at the peripheral nerves, if we look at the cranial nerves, are the neurons touching each other? No. So just like with skeletal muscles, I don't want my skeletal muscles touching, because if electrically something is happening on one and they're touching, what's going to start happening? Electrical events on all of the muscles that are touching, right? So again, if I have information coming from one neuron to the brain, do I want it to be touching and make something happen? No. Do I want that information potentially, again, this little spot's being triggered, do I want the information to be also carried over here? No. So I need my nerves, just like my skeletal muscles, to be individually wrapped. And then to help again, that cranial or spinal nerve, when it exits out, it's going to potentially send nerves to a bunch of different locations. So then I need them grouped in units and wrapped in connective tissue. And then the whole nerve needs to be protected. So I have endo, peri, and epineurium is what I call the connective tissue. Okay? So, in the peripheral nerves, there are layers of connective tissue associated with neurons and groups of neurons. The blank is the middle layer and consists of connective tissue wrapping around a collection of neurons called a fascicle. Perineurium. Again, on our neuron model, it shows that gray material wrapping around the Schwann cell and the axon. What would we call that? Endoneurium. Okay? If you look at the eyeball, and if you look at the yellow nerves, and there's a little bit of white around them, what would that little bit of white coming off the eyeball? That would be the epineurium. Okay? So again, you have different layers of protection. All right. Spinal cords, anterior or ventral horns, tend to be synapses between neurons. These neurons tend to be associated with which of the following? So, anterior tends to be what? Sensory or motor?
Motor.
Motor. So, if the anterior is motor, what does the back have to be? Sensory. So, if I inject my spinal cord in the back with some numbing, am I numbing my ability to walk, or am I numbing my ability to feel? Feel. And that's what an epidural is. So could people potentially with an epidural still kind of walk around? Yeah, but they can't feel anything, so are they going to have that feedback to their brain of I'm moving my leg and I'm feeling the wind and I'm feeling the pressure and so that's the damage of why we don't want people on epidurals walking around. Okay? All right. The test for function on this cranial nerve is to test the gag or swallowing reflex. Cough and have the person speak. So we're in the back in the pharynx, and we're moving a little bit into the larynx. So what's the best answer? Glossopharyngeal. Okay? The trigeminal was the one that a bunch of people picked. The trigeminal has big parts out here. So the trigeminal is going to be more associated with the teeth. Okay? The teeth and numbing the teeth so you don't feel as I drill into your teeth. But the back of the throat is more about glossopharyngeal. Okay? All right. The blank area of the brain is responsible for controlling blood pressure, heart rate. Most of you got this. It's the vagus. And you know it by name. And what is the vagus by number? 10, which is the letter X. Because you have to give them to me in Roman numerals. Okay? The blank area of the sorry. I jumped questions. So, the blank area of the brain is responsible for controlling blood pressure, heart rate, and breathing rate. That's your medulla oblongata within your brainstem. So, what nerve is probably coming out of it? The vagus nerve. Okay? All right. The part of the brain that is important in controlling and regulating posture, fine-tuning conscious and subconscious movements. Not overshooting. Okay, what is that? Cerebellum. All right. Deciding to move is your frontal cerebrum, but making sure as you reach, you don't overshoot, you don't undershoot, you don't go too much to the right or left. A lot of that's the cerebellum. Visual information is primarily headed to the primary visual cortex of the cerebrum. Which lobe? Occipital. Rods and cones are special photoreceptors within the retina of the eye. These cells will synapse with a first-order neuron within the retinal layer. These first-order neurons are special because they're only ones in the whole human body that we associate as being bipolar. So their little cell has two extensions coming off of it. Okay? The blank is the arm of the autonomic nervous system known as thoracic columbar division. It is characterized by short preganglionic fibers and long postganglionic fibers. Sympathetic. Okay? An example of a cholinergic receptor. What does cholinergic mean? Cholinergic, what's the neurotransmitter we're talking about? Acetylcholine. So, a acetylcholine receptor, that's what I'm telling you. If I said an adrenergic receptor, what neurotransmitters am I talking about? Norepinephrine and epinephrine, the catecholamines. Okay, so cholinergic receptor that might be found on postsynaptic membranes of postganglionic neurons, and it's in both the sympathetic and the parasympathetic system. What receptor am I asking you about? Which cholinergic one? Nicotinic. And remember we kind of talked about it got its name because it responds to nicotine. Okay? All right. In the central nervous system, myelinated axons would most likely be found in all the following locations except, is the corpus callosum white or gray matter?
White.
White, and so that is where we expect to find myelination. Is the dorsal column, the anterior column, or the lateral columns, is that white or gray matter? That's white. Okay? Is the arbor vitae white or gray matter? White. So, which of these is not white? The posterior horn. What is the posterior horn? The afferent sensory neuron is ending, and some inner neuron's little one-eyed monster dendritic head is starting. All right, so synapses are occurring there for sensory information. Okay? Which of the following parts of the inner ears where static equilibrium otolith organ is contained? That's in the middle, so it's not the cochlear and it's not the semicircular canals. That's known as the vestibule. Right? Interocular pressure is determined by circulation of blank in the anterior cavity of the eye. Aqueous humor, if I have too much, what disease am I eventually going to get? Too much aqueous humor? Glaucoma. Glaucoma. Okay? Which cranial nerve is assessed with the pen light? Well, didn't I ask this question already? So it's oculomotor again. Right? See, I gave you a good freebie there. Which cranial nerve has control of salivary gland secretions along with lacrimal gland tear secretions and can test the function of the lacrimal gland with ammonia fumes? Facial, right? And the reason why it's the facial is because of that tie back to your lacrimal gland secretions. Okay? Which cranial nerve is associated with the large portion of the parasympathetic? That's the vagus. I did that one earlier, right? Which cranial nerve is associated with control of the sternocleidomastoid? The trapezius. Okay? And you test it by people having like push up when you're like pushing down on their shoulders. Accessory, right? Y'all said that? Okay. The epidural space of the spinal cord, which is location used for many medical procedures of drugs. Which layer of the meninges? It tells you epidural, so it's the dura mater. A viral disease that causes damage to part of the spinal cord leads to muscle weakness and or paralysis. So what part of the spinal cord did it kill? Not the front.
What? Oh, the
Anterior or ventral.
My bad.
If you put front, that's wrong. It's not the correct anatomical term.
Good thing it's multiple shoulders.
Nocioreceptors. Nocio, think pain. So what do endorphins numb? Nocioreceptors, right? Endorphins compete with that. Visual pigments. So most of you put rhodopsin here, but what is rhodopsin? What do I break rhodopsin into? Retinol A, vitamin A, and opsin. Okay? And again, if you switch them, it screwed it up. If you put rhodopsin, I just gave you half the credit because I'll give it to you for opsin. And then how an action potential is propagated along the axon is dependent upon the axon's myelination or unmyelination. Which of the following is a type of conduction or propagation scheme where the axon is myelinated? And so it jumps. That is salt, remember? Saltatory versus continuous. Heel to toe. All right, any questions? What? Any questions? Okay. Questions from you guys online? Do you have any more in the chat? All right, well, I'm going to stop the recording. That is all we